The purpose of this protocol is to evaluate in an in vitro model whether artificial tear formulations can protect human corneal epithelial cells from desiccation.
This method can be used to help identify dry eye formulations that can aid in ocular protection for individuals with dry eye symptoms.
This assay utilizes a very sensitive measure of detecting corneal epithelial cell metabolic activity. As a result, small changes in corneal cell health due to desiccation can be detected. Demonstrating the procedure today will be Parisa Mirzapur, Nijani Negarut Kamaran, and Adeline Suko.
Begin by growing immortalized human corneal epithelial cells in collagen-coated flasks with 20 milliliters of DMEM-F12 containing 10% fetal bovine serum and 1% penicillin streptomycin at 37 degrees Celsius and 5% carbon dioxide, changing the media every 2 to 3 days. Once the cells are almost confluent, remove the cell culture media and add 4 to 6 milliliters of cell dissociation media to each flask. Incubate the cells at 37 degrees Celsius until they detach, periodically checking them under the microscope. Add 2 to 6 milliliters of DMEM-F12 with 10% FBS to each flask, and transfer the contents to a 50 milliliter centrifuge tube. Centrifuge the cells at 450 to 500 times G for 5 minutes, then aspirate the supernatant and resuspend the cells in pre-warmed media. Determine the cell concentration with a